Hi friends, today I am here to talk a topic of or teach a topic of agglutination reactions. As we have discussed in the previous videos that antigen antibody reactions are of two types. One is precipitation, second one is agglutination. What is the definition of agglutination? Suppose if an antigen enters or present in a sample of a patient or a client, it undergone taken into a test tube with an antibody present on it. By adding antibody, these both form a clump. These both antigen antibody form like a clump like this. This is known as a agglutination reaction. Main result of an agglutination is clumping of a sample or a blood sample. It may be a blood or a serum. Next, antigen occurs between agglutination occurs between antigen and antibody. The antibody which perform an agglutination reaction, antibody is known as agglutinins. The antigen which perform the agglutination is known as agglutinogen. Agglutination reactions are several types. First one is a slide agglutination, second one is a tube agglutination, third one is a heterophile agglutination, fourth one is a comb agglutination. Comb is further classified into two types: indirect, direct. Indirect combs classific agglutination, direct combs agglutination reactions. Latex agglutination, passive agglutination test. These are the main types of an agglutination reaction. Now I am going to deal in slide agglutination. What is actually a slide agglutination? Slide agglutination test. Take a test tube with 4 wells. Slide with 4 wells. In first one, suppose it is in a first one, second one, third one, fourth one. Add a positive control reagent in it. Second one, negative control reagent. Plus saline water. Plus saline in it also. Here sample plus saline. Here and add if a sample contains an antigen, here we are going to add an antibody. These both get involved in a reaction such as an antigen antibody reaction and form a clump in it. This is a slide agglutination. Next one is a test agglutination, test tube agglutination or test agglutination. Here what is actually mean of a test agglutination or a tube agglutination? Take a series of test tubes. Add an antibody in equal antibody 2 concentration 2 right, and different concentration of antigen like 1 2 antigen 3 4 5 equal amount of antigen antibody get clumps here these both are get clumped. Why we are going to say clump means the antigen concentration and antibody concentration are both are equal in the, suppose label these tubes as a B C D. Here in the B test tube the concentration of an antigen and antibody are equal. So these both are get clumped tightly or very darkly. Here these both are unequal concentration or in unequal concentration. So only some of the clump can be seen in this all of this. Here the excess of an antigen is takes place. Here excess of an antibody takes place. This is a tube agglutination. Next topic is next type of an agglutination reaction is heterophile agglutination reaction. Here what is mean by heterophile antigen? What is mean by heterophile antigen means type of antigen. It is a one type of antigen in two different species but same epitope. It means it is a type of one antigen present in different species but same epitope. Suppose it is an antigen. These are epitope. Belongs to another A species. It is belongs to A species. Here it is an antigen which shares the structure of epitope is same with a B species. It is a heterophile antigen. These both are different species but consisting of the same epitope. Then A species get reacted with an A antibody. Suppose A antibody. B species also react with a B antibody. And as both of these are consisting of a same epitope or sharing a same epitope, these can cross react with a B also. This also. It can also cross react with A also because both epitope are same but species of an antigen is different. Here what is different? It means species only different but epitope is same. This epitope is a part or a region where the antigen come and bind with an antibody is known as a epitope. Where it bind means paratope of an it is a region of paratope of an antibody. This is the main introduction of an heterophile. Your example of an heterophile reaction, it means Weil-Felix reaction. It going to recognize a typhus fever diagnosis for a typhus fever. Antibody present. It shows a antibody present in serum of rickettsia. It can cross react with a Proteus with a bacteria which is going to cause an infection. Here next one is a Combs agglutination test. It is also known as a antiglobulin test. It is discovered by or performed by Combs, Morant and Race. There are three people totally: Combs, Morant and Race. By dedication or a work by these three members, the Combs test is discovered. Combs reagent is a anti-human globulin. The reagent which is used in a Combs test is known as a anti-human globulin reagent. As we have discussed in a previous that Combs test is classified into two types. First one is a direct and second one is an indirect. Let us discuss what is actually a Combs direct test. Here plasma it is used to detect the antigens present on a plasma cell is a direct Combs test. Here suppose a patient sample is taken. here and wash it three times. Wash 3 times with a saline water and add a Combs reagent. Here, what is a Combs reagent? Anti-human globulin is a Combs reagent. Combs reagent is added. Here it forms a, suppose it is in a plasma cell, it forms like this. Like this. By examining the solution after the adding of a Combs reagent, here what we can see that a clumping of a solution, so it is known as a Combs direct test. Next one is a indirect Combs test. As we have discussed in the previous, that Combs are classified into how many types? 2 types. First one is a direct Combs test, second one is a indirect Combs test. Then what is actually a mean of a indirect Combs test? Indirect. It is an indirect. It is going to detect, or it is used to detect a antigen present in or a present freely in the body. It seems is known as a indirect Combs test. It is going to detect a freely present antigens. Here, examples of indirect is hemolytic anemia, erythroblastophitalysis, RHO group, ABO group. These are the examples of an indirect Combs test. What is a procedure? Let's discuss what is a procedure of an indirect Combs test. Take a sample of serum which consisting of an antibodies. Add a serum into test tube of plasma. Here plasma is get added. Wash it. After washing, here we can see the clumping by examining another microscope. These are the indirect agglutination test. Next one is the latex agglutination test. Here these are the latex molecules or substances. Here these are an antibodies. Antibodies and latex are added. Here the formation of a latex attached with an antibody and adding of an antigen forms agglutination. Like this, suppose it is a latex molecule, antigen. Suppose it is an antigen. It is epitope, it is a paratope. It is a antigen. Epitope, paratope. Antigen. Epitope, paratope. Next one is a what is actually a use of latex agglutination means. It is going to detect a Streptococcus antigen. It is going to detect the anti-streptococcus antigen. Here, next one is a coagglutination reaction. Here, the cells covan one strain cells of Streptococcus aureus are added with an antibody. Here forms a complex of both of these, and adding of an antigen, it is an antigen. Here the formation of an agglutination. Suppose it is a covalent cell. If it is an antigen, it is an antigen. Here it is a epitope. It is a paratome. This is the agglutination complex. It is a clump. It is also a clump. What is the main property of an agglutination reaction is formation of a clump. Okay friends, thank you for watching. Please like, share and subscribe to my channel.